So one of the keywords of my talk today is about small molecules. So what are small molecules? If you search the Google, you will probably getting different answers. Some say a molecule with less than 900 Dalton. Some say between 100 and 1100 Dalton. Well, we don't have to argue about that. In medicine, a small molecule drug is referring to a chemical entity of low molecular weight, in general below 1000 Dalton, which is applied to patients with the intention to heal a disease or prevent a secondary consequence of the disease. And if you search the web of science, you will see majority of the outputs are chemistry related research. No doubt, small molecules are also important in medical research. Here I take one of the highly cited paper, for example. In vivo activations of the p53 pathway by small molecule antagonists of MDM2, published in Science year 2004 with more than 4,000 citations. The small molecules are referring to the natrine 1, 2, and 3 here. As you can see from this slide over here, they are molecules specifically designed to inhibit certain proteins, such as kinases, in the cells that have been implicated in disease. The little graphic shown in figure B, C, and D here are actually referring to the interactions of the molecule with the protein, in this case, MDM2. MDM2 is a protein that binds to and negatively modulates the p53 tumor suppressor protein. The concept of interaction between the small molecules and a protein is analog of key and lock principle. You got to have the right key to open the lock. Now, imagine this is a schematic diagram of a lock where you have 3 different sites of interactions. Here, here, and here. That resembling the characters of a lock. And your key cannot be too small. Otherwise, it won't fit well to the lock. Okay? Similarly, your small molecule cannot be too small. Otherwise, it won't interact well with the protein. In medicinal chemistry, we can design the small molecule so that it not only fits well to the site of interactions, but also interacts specifically to one interaction site. As you can see here, the benzene ring interact with each other while the van der Waals bond, and we can build the molecule with additional moiety, in this case, an acetyl group, to engage a hydrogen bonding with this neighboring amino acid residues, or this methyl amino group to engage a strong ionic interaction with this particular red color residues. So after all, we have a nicely fit small molecule that can either turn on or turn off a function of a protein. Today, small molecule kinase inhibitors are being intensively pursued as new anti-cancer therapeutics. And there are approximately 80 inhibitors have been advanced to some stage of clinical evaluations. Here we have 2 well-known small molecule drugs, baricitinib and remdesivir. Do you know what these drugs are used for? Baricitinib is a Janus kinase inhibitor, and the remdesivir is a ribonucleic acid inhibitor. Together, they are currently being used to treat COVID-19 patients. Many of you may wonder how small is a small molecule. In terms of size, a small molecule is typically in a few nanometers in size or less. It is so small that we can't really see it. Perhaps with atomic force microscope, we can see a real molecule like this naphthalene tetracarboxylate diimide. This photo credits to P. Moriarty and co-workers. They have done an excellent research on it. Small molecule drug discovery and development. The process of small molecule drug discovery and development can be complex and challenging. A typical workflow begins with screening of compounds. Ideally, it should be a pure chemical entities. But we also work on crude plant extract or fraction. Next, we may identify a heat compound which show promising bioactivity. It then follows heat to lead and the lead optimization. And finally, carried through the preclinical development. All these processes are lengthy, resource demanding, and require multidisciplinary teamwork. We cannot work alone. In academia, I think, we are still unable to carry out all these processes till IND, the filing of investigational new drug. I mean, in the context of Malaysia. What happened in the past, we show our lead compound to the biotech or pharma company, and we get some milestone payment in return. The development of small molecule drugs has never been easy and straightforward. Here I share a case study. During the heat generations for a serine protease, we came across a crystal structure of the protein in complex with a small molecule called benzalmethine. So we rebuilt the structural model by using the PDB ID obtained from the protein data bank. If you still remember the site of interactions, here the benzalmethine is too small to be fitted well into the site of interactions. And there are plenty of room to expand. So next, we design new molecules based on benzalmethine core structures. We introduce additional functional groups such as these, and this. And we synthesize about 200 libraries. And from this pool of libraries, we identify an initial heat compound showing Ki value of 2.9 micromolar. And subsequently, the initial heat molecules were further optimized through a series of structural transformation to give a heat molecule of 40 nanomolar in Ki value. Another challenge that we often met is the selectivity problem, such as the selectivity of a target kinase in this case. We have compound 1 which show very potent inhibition against ALK kinase, 5.6 nanomolar. But compound 1 was also very potent against Aurora A kinase activity. So this is not good because the undesired inhibition of Aurora A may have an implication in toxicity, especially a cardiotoxicity. So how? So we have to check the interactions of compound 1 with Aurora A protein structure. At the site of interactions, we can see the second nitrogen of the compound 1 interact with Aurora A protein amino acid residues. Here is the neighboring lysine 162 in a shorter distance as compared to ALK, which is not shown here. That is the reason why compound 1 show good inhibition against Aurora A. And to solve this problem, we have to disrupt the interaction. We can do this by introducing dimethyl groups into this pyrazole ring. So it prevents the second nitrogen from interactions with the lysine 162 by steric hindrance. At this stage of development, we try to avoid big change in the structure. Otherwise, the resulting new structure would not fit well again into the site of interactions. And we have to depend on the availability of the starting material and the easiness of introducing a new functional group at this late stage of development. So understanding the structural basis of kinase inhibitor selectivity is crucial to the ultimate goal of developing selective inhibitors to target every member of the kinome. So what have the small molecules to do with nanomedicine? So what is nanomedicine? Nanomedicine is the medical applications of nanotechnology for diagnosis, treatment, and management of human health. This field is very broad and multidisciplinary, involving chemistry, physics, material science, molecular biology, pharmaceutics, medicines, and clinical translation. In a simplified term, nanomedicine can be defined as a medical applications of nanotechnology. Nanomedicine for accurate cancer imaging. Iron oxide nanoparticles are one useful tool against cancer. When these nanoparticles are engineered with a specific coating, such as targeting ligand, folic acid, they bind particularly well to the tumor. Their magnetic properties make them suitable imaging agent for MRI, while their size and concentration in tumor allow a very high resolution and an accurate mapping of lesions. Surgeons can thus rely on this imaging to select a proper plan to remove the tumors. Nanomedicine for targeted therapy and drug delivery. In therapy, nanotechnology is at the forefront of both targeted drug delivery and intrinsic therapy. For instance, nanoparticle can already be injected into the tumor and then be activated to produce heat and destroy cancer cells locally, either by magnetic fields or near infrared. Meanwhile, the encapsulations of existing chemotherapy drug allows localized delivery, therefore reducing the quantity of drug needed by the patient and the side effect on healthy tissue. In this schematic diagram on the left, a drug-loaded multifunctional stimuli-sensitive drug delivery system. Drug A and B can be loaded into a nanocarrier such as liposome or polymeric micelle. And depending on the purpose of the drug delivery system, various agents can be added to the nanoparticle to target the drug delivery system to a particular tissue, to increase cell penetration, to enable imaging, or to release the drug in response to a given stimulus. In nanomedicine, our interest is the functionalizations of drug and nanocarrier, control release study, and drug delivery. In this example, we demonstrated that PLGA nanoparticles formulation with triorganotin carboxylate show a much greater tumor growth delay than the free drug. Our group is interested in making small molecules. Organic chemistry is still one of our core business. We are interested in developing new methods for the preparation of small molecules, especially the biological important small molecules. What we do, we design new and efficient ways to a variety of different derivatives. Here are some examples that we worked in the past. We collaborate with academia as well as pharmaceutical industry, and most often we make the molecules and did the biological testing. The second main research pillar is medicinal chemistry. We are interested in designing new potent molecules. We work from the designing stage all the way to heat and lead optimization. And the aim of our work is to refine each heat series, aiming to produce more potent and selective compounds with desired pharmacokinetic properties. In this connection, we have collaborated with many researchers. We have two, and many of them are from local and abroad. We are also interested in natural product research. As you may know, Malaysia is one of the 12 mega biodiversity countries, and we have access to a diversity of flora and fauna with over 15,000 species of flowering plants, herbs, and medicinal plants. And natural products are a rich source of chemical entities for pharmaceutical-led and novel compound discovery. However, research in this field is not easy. Often the secondary metabolites that we have isolated are minute in quantity. And We are actually the first group to tackle the scarcity of a dengue virus inhibitor, Panduradin A, which was originally isolated from a ginger species. Today, I'm going to share with you some of our latest research outcomes. We have recently discovered a way to selectively introduce a hydroxy group to anthraquinone, which could be potentially be used as an electroactive species in organic redox flow battery. Studies have shown that functionalization of anthraquinone with electron donating hydroxy group has led to a lower reduction potential and a better aqueous solubility. So a little bit background of the study. Redox flow batteries are large-scale devices for energy storage and conversion. It solves the mismatch between the intermittent supply of renewable energy resources and their variable demand. So these rechargeable electrochemical energy storage devices utilize the oxidation and reductions of two soluble electroactive species for charging, which is absorbing energy, and discharging, which is delivering energy, as shown in this schematic diagram. The electroactive species are the key component of the redox flow battery. And currently the method for direct hydroxylation of the anthraquinone is not available. And most of the anthraquinone discovered so far were either prepared from multi-step synthesis or rely on natural product isolations. And these methods suffer from low yields, hindering further development of anthraquinone for their uses as electroactive species in the redox flow battery application. So in this work, our aim is to develop a new method for direct hydroxylation of anthraquinones. Specifically, the hydroxyl group will be introduced at the auto opposition adjacent to the carbonyl group, which is shown here, 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 or here. We use inexpensive reagents while CH activation approach. So far it works for the following substrate. And we are still working on many other anthraquinone derivatives. Hopefully we can publish the paper soon. All right. So I would like to share with you some of our experience with the pharma industry. During a project collaboration with the Origin Discovery Technology, a pharma company from India, our students get the chance to work together in developing a few new drug candidates for preclinical studies. An outcome from this collaboration include journal publication and patents. Last but not least, I would like to thank NanoCare for organizing this seminar. The aims of this webinar program are to enhance the intellectual atmosphere of the university, to disseminate faculty's expertise, and to connect our researcher with scholars and experts from local and abroad. At the seminar, our students and researchers come together to keep track of the recent development and to help develop ideas and collaborate. Lastly, I would like to thank you for your kind listening and your kind attention. So thank you. So now, open to the floor if you have questions. Do you have questions? Can you hear me?